I learn all of the new products that come in that our company creates. And then I get to kind of create a lesson plan like your teachers do. And then I teach it to our customers and the people internally. And I love it because I get to learn new things every day, new things that are happening in biotech. And then I get to share it with my colleagues and our customers. It's a really fun job, and I love that I get to learn every day.
That's super cool. So what kinds of like products, what does Illumina do as a company broadly?
So we make quite a few things, but what we're mainly known for is we sequence DNA, and we make the solutions that we use to put onto instruments that we make as well to find out what our DNA is about.
Very cool. Thank you. Elizabeth, do you want to introduce yourself?
Yeah, hi, my name is Elizabeth Bottenberg. I am a quality control analyst, and as Sarisha said, we put solutions into these big sequencing machines to make sure that we can actually sequence the DNA. And so it's my job, before we sell those solutions to other companies, I have to make sure that they are up to standard. And so I often in the lab testing those reagents and making sure that they're good to go before we package them and send them off. And the reason why I love this job, Illumina was my dream company when I was in high school and going through, and always being part of the gene project was a big thing for me. So finally making it is a big deal for me. And I learned so much from my other colleagues and my manager. And I love that it's a continuous learning experience.
Very cool. So this question's for both of you. So when we talk about sequencing DNA, first of all, why would we want to do that? Like, what do we gain from knowing the sequence of DNA?
Do you want to answer, Elizabeth, or do you want me to get started?
Can you repeat the question one more time?
Sure. What's the benefit of knowing the sequence of DNA?
Ooh, depending on whose DNA you're testing, we can learn a lot about maybe if you're susceptible to different diseases. We can see if your family history has a pattern of certain qualities or traits that you may also possess. I know for sure we use gene sequencing for family planning. So if you plan to have kids one day, what would your kids look like, or what kind of gene history will they have, depending on what you have and what your parents have. And so that's a big effort behind gene sequencing.
Super cool. Thanks. And so you mentioned that you are looking at the quality and making sure that reagents work well for the scientists who then go use them and maybe do DNA sequencing or any various other numbers of things that you produce. What exactly is a reagent? I don't think I learned the word reagent maybe until I was in college. What does a reagent do?
Yeah, and a reagent's a fancy word for the solutions that we'll put into our sequencing machines to make sure they work. And so reagents can be liquids, they can be solids. A cool part of the DNA sequencing process is that we use these little tiny beads, and by using ethanol and different wash solutions, we can actually have the DNA stick to these little beads so that we can eventually sequence them and read them.
So for those of you at home, you might be thinking of beads as something that you might put on a necklace, or beads that you might even see. And this is actually perfect. It's Indigenous Peoples Day. So happy Indigenous Peoples Day to everyone at home. In Indigenous bead artwork, that kind of bead, these beads are so small you can't even see them. They're teeny, 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 tiny. And a lot of times in molecular biology on the whole, that's, if you just look at a scientist doing molecular biology, what it might actually look like to like the human eye is just people moving clear liquids from one tube to another, because the really important stuff that's happening is super, super tiny. And so a lot of times we'll have like a mix of stuff. So back when I was in graduate school, I worked on squid. And so a lot of what I did, I would basically be taking a squid sample or maybe a bacteria sample, and I would squish it up, and then everything that's present in cells would be in that like smoothie of squid. Now, how am I supposed to figure out what's going on in really tiny specific interactions if I have everything that exists in a squid? That's like a lot of stuff. So sometimes what scientists do, they need to basically have like a magnet that pulls out the specific thing you're interested in. And sometimes you'll connect a thing to a bead that will help you pull out the important stuff. If you've ever had one of those like etch-a-sketches that like pull magnets to make a picture, it's sort of a similar thing. So you'll have a bead and then a thing stuck to a bead that pulls out the thing that you care about. And then sometimes these beads are even magnetic to like pull the thing that you're interested in to the side of the tube so you can get rid of all that other squid stuff or whatever it is that you're working on. You could be working on cheetahs, you can be working on bacteria, fungus, whatever. And so that's the kind of stuff that Illumina makes. And if they didn't make it, we wouldn't be able to do all the complex stuff that we do. If you have any questions at home, please submit them to the Q&A or alternatively into the chat. Either one works totally fine. My next question for you two, what kind of education did you need to get the career that you have now?
So I got my bachelor's from UCSD and I got it in biochemistry and cell biology. And so right out of college, I went and worked for a small startup before I joined Illumina.
Yes, same. I also went to UCSD and I majored in biology and I had a special concentration in global health. And I definitely thought about grad school and maybe it's still in my plan. But before I got to Illumina, I was actually doing COVID testing, still in the lab, working with some pretty gross samples and doing the processes to make sure that to see if people tested negative or positive.
Very cool. So a really important thing that we just learned is that you don't necessarily have to go to graduate school to have a career in science. You just don't need it. Like sometimes you do. If you want to be a scientist working and running your own lab, you need a PhD to do that. But if you want to be involved in science, you don't necessarily have to be in school for as long as I was in school, which was approximately forever until I was like 30. And you don't have to do that. That's not necessary. So that's great. So what would you say is for both of you, what is your favorite part of your job and your least favorite part of your job?
I guess my favorite part of the job, which I guess I told during my introduction, is just I get to learn something new every day, which I don't think a lot of people get to say when they've been working at the same company for 5 to 10 years. And the thing that I don't like, which I think is just with any job, is just, documentation or all the meetings that you have to go back and forth with. I obviously love science and I'd rather just be in the lab and learning all the new scientific stuff, but any job comes with all the paperwork and everything that you have to do.
I think emails is the worst part of every job, no matter whether you're a scientist or an electrician or like emails is the worst part of any.
Yeah, emails and phone calls.
What about you, Elizabeth?
Yeah, I have to say the same. When I came to the job and even my last job, often you learn your one task and that's all you have to do. But with Illumina, they really want you to learn everything you could ever possibly learn. You can learn from the bioengineers and learn more about how do the machines even work, even though that's not part of your job. You can learn more about the planning process. When are the samples coming in and why is it important that we meet certain deadlines? And the least favorite part of my job is, like you said, usually we're moving clear liquid to other clear liquid, and sometimes it's really hard to know that, oh, yes, I'm doing what I need to be doing. Because oftentimes we'll be doing all of our tests and then we'll go through a long sequencing process and we have to pray for a pass or a fail. And so I think the hard part sometimes is having faith in the science process to make sure that you really are doing everything you need to do to make sure you get the results you need.
Totally relate to that too. All right, we have a question from Annie. What is DNA versus RNA? And then on top of that, we've heard a lot this year and last year about mRNA. And then what's mRNA? So let's go through DNA, RNA, and mRNA. And then we have a follow-up question to
Okay, I can get started, I guess. So DNA is basically it's a double helix code for what makes up who you are. It kind of tells your body what your eye color is, what your hair looks like, how tall you're going to be. It just basically is the code of who you are. RNA is a single strand and it translates for the proteins, all the stuff that kind of works inside of your body. And then mRNA is a type of RNA that's just messenger RNA.
Yes. So DNA is like, I kind of like to think of DNA as like the reference book in the library. Like you don't take that out of the library, don't tear a page out of that. It's like it's the everything book. It has everything. And then RNA is like what you're using that day, like the thing you take a photocopy of. That is like what you need to make that day, basically. And then mRNA, yeah, exactly. It's a type of RNA. And mRNA vaccines are basically using mRNA. So mRNA stands for, just like Sarisha said, messenger RNA. And so that's like the photocopy that you took out of the reference book to then go like make your recipe for the day. And we are putting in ourselves that recipe and telling ourselves, hey, make this thing. And then our bodies find that thing and say, okay, that I need to get rid of that. And so you're basically instead of taking the COVID particle or the virus, and putting it in our bodies, you're making your body make a little piece of the virus that then they can create attacking particles against. So that's what that's about. So your next question is, does Illumina just work on DNA, or are they also working with RNA and mRNA?
Yeah, we work with RNA as well. So we use those solutions or reagents if we're working with DNA or RNA. And then we translate the RNA into DNA so that we can put that onto the sequencer. So it's any type of input, DNA, RNA, and then we're able to sequence that.
Great, thank you. We've got a question from Mrs. Young's class. This is Alexa from San Diego, same city as you. Was there someone in science or biotech that inspired you to pursue this career? Was it a family member, a scientist, a teacher, or a professor?
I think for me, my own family genetic history really pushed for me to be in science and learn more about genetic history in general. But I think the one person that I owe a lot of my own inspiration to was when I was in a research lab in college and I had a postdoc, Guillaume, and he was like one of those first people that said, oh, you know what, you really can do lab bench science. Like, I really can see you as a great scientist. And so I really owe it to Guillaume that he really pushed me to see my potential.
That's awesome. It's good to hear. I'm sorry, did both of you get a chance to answer that question?
Oh, I'll just answer real quick. For me, it was when I started learning in my AP biology class in high school. I think that's when it really clicked to me about like what is going on inside of our bodies and how cool that is, especially when we talked about genetics. And then once I went to college and took a genetics specific class, I just wanted to learn more and more and start working on it. So it was really nice that Illumina is in San Diego, where I was going to school. So that became my dream company after that class.
Awesome. So obviously not all of our viewers are in San Diego, but there are biotech companies all over the country. There's a ton in Boston. There are some in Philadelphia. And admittedly, those are the two places I've lived. So those are the ones I'm familiar with, but they're all over the place. There are some in the South and in the Midwest and everywhere. Does Illumina only have offices in San Diego, or are they all over the place?
We're global, we're worldwide.
Right.
Yeah, I know there's another Illumina campus in Tennessee, but they're, I think maybe in Kansas and all over China and Singapore and the UK. I don't know if there's really a country that Illumina's not in.
Yeah, and we even have what we call field-based teams. So wherever you live, you could be a scientist and help out the other companies that are near you. So you can go give them a training on Illumina, how to use all of our Illumina products, and you're still working for Illumina where you live.
Yes, awesome. Sounds good. We got a question from Ina. What are some research projects that biotech would tackle? Like, what are the, could you give us an example of maybe a project that you would supply reagents or, supplies for, or big society-wide problems that your company kind of helps with?
Sorry, I think you cut off right at the end.
Oh, sorry. Basically, like what are some specific examples of projects that biotech tackles?
Yeah, it's a pretty broad range. I guess some of the examples that Illumina works on, we try to tackle the big problem of cancer. So we work with other companies that maybe create certain medicine or certain therapies that are trying to target certain cancers. So we work with them to provide how to detect if you have a certain cancer, and then if it does show up, then they can give you the medicine that will help hopefully get rid of the cancer. But we also work on agriculture stuff. So maybe if there's something wrong with the wheat that you're growing and you want to find out why, we also have reagents or products that help with that too.
Very cool. So one of the people who works for Skype a Scientist also works for another biotech company, AstraZeneca, and that ties into a lot of what you were saying. And I wouldn't be surprised if the source data that he's working with didn't have some Illumina reagents involved. This is like, science is very collaborative, and a lot of different biotech companies can go into one project. So one of the things that he's working on is trying to figure out, why would one cancer drug work well for one person who has lung cancer, but not for another person that has lung cancer? And this all really comes down to what changed in the cell that made it cancerous, that went from a normal lung cell, just a standard lung cell doing the respiration thing that lungs do, and then turned it cancerous. And so what a scientist might do is take a tumor and then sequence the DNA of the tumor, and being able to look at that DNA and then see, like, let's say we have 100 cancer tumors and that we have the DNA for, and 20 of those all have worked really, really well with one drug. Like, one drug totally knocked out 20 of those 100, and they all have this one unique sequence in them. We can then find a new cancer patient and say, oh, you have that sequence that worked well with this drug. And this is called personalized medicine. And so this is applicable to lots of different things that aren't cancer related, but that's why sequencing can save lives. It's really, really important. So thank you for that. We've got a question from Mrs. Young's class. Is the field of biotechnology growing? And if so, what's responsible for the growth in this field?
Oh, absolutely. It's growing all the time. I think when I was in high school, they're like, wow, this is a new big thing. And I think it's partially because of personalized medicine, there's just such a need for every niche of biology to be able to explore what's out there. And why is it growing? I think because there's just so many, with the rise of technology, there's just so much more interest in certain specific areas of science that we need somebody that only works on that one lung cancer that's really stumping scientists around the world, and it's never just a one-person project. You always need a team to be able to figure out our world's greatest problems.
The more, this is like a scientific thing that happens with all science, whether you're studying, giraffes or space or whatever, the more you know, the more you realize there is to know. Like, every time you get an answer in science, 3 more questions pop up. And so that's kind of like the age-old reason science is always growing, because there's so much to know, it's impossible to possibly know it all. So awesome.
Yeah.
Thank you.
And I just wanted to add too, I think it's also growing, like Elizabeth mentioned, because of technology. And to be a scientist, you don't have to be working in the lab pipetting all the solutions all the time. We also have people who are like bioinformatics scientists. So they're working on coding so that we can do data analysis and find out more about what's going on in the reagents or in the DNA sequence that we're finding out about. So because our technology is getting better, we're finding out all these different avenues to grow biotech itself. So there's more jobs that way as well.
For sure. You touch on a really important point, and that's the importance of computer science in biology broadly. We had another question here from Gavin. Is there anything in this process to do with chemistry? And yeah, biochemistry is so, so important and really like at the heart of why a lot of these things work. And so, like we mentioned, like science is super collaborative. Like one person can't solve big problems. You need a lot of different expertises working together at a company like Illumina or at an academic lab or wherever to really get to the heart of things. So is there a place for chemistry and biotech? Yes. Also, if you are watching and you are thinking about maybe learning how to code, please do it young. I tried to learn when I was 27 and it was very challenging. So learn coding as early as humanly possible. A follow-up question for you two, what branches of chemistry might be useful in biotechnology?
All of them, I think. It really, whatever you're interested in, you will definitely find that there is an application for it in biotech. Like I mentioned, I have a biochemistry background, and you kind of need to understand, I mean, chemistry and biology just go hand in hand. So you kind of need to understand how the chemistry works to know how the biology works. So any and all the branches of chemistry are totally applicable in biotech jobs. Yeah.
Yes, for sure. Do you have anything to add, Elizabeth?
No, and I mean, the assays or the processes that I do in the lab, it was not because of a biologist that knew that DNA could bind to beads. That was a chemist that knew, oh, the way that these structures work, it's because of the way these beads and the DNA interact that I can actually do my job as a biologist. But it wasn't without a chemist for me to do that.
And biophysicists also are super helpful for like designing like the magnetic beads or any number of machines that allow the chemists to do the work that allows the biologists to get the answer. So it's, like I said, it's super interconnected. And yeah, that's how science works. The question, we got another question. This is, we're an elementary school class. What skills could students in 4th and 5th grade be working on now if they'd like to pursue a career in science one day?
That's a good question. I think, I mean, I'm sure you guys are probably already kind of working on this, but if you just like observe how things work around your house or around school or outside, just kind of getting to know how things work and how things interact with each other, that's just kind of the basis of what we do and how we learn. It's just on a more smaller and tinier level, as Sarah was saying, that we can't really see how it's working. But if you try to understand how things work around you, you're already kind of acting like a scientist.
Mm-hmm. And I've learned a big part of science is that it is people oriented. And like we said, you can't just do science alone. You have to have your friends in the lab with you. Oh, this didn't work out, like help me troubleshoot or figure out what went wrong. And so I think a big part of being a scientist is being open to collaborate, open to making friends, and being comfortable with being uncomfortable, and knowing like it being okay if you don't know something. There's so much that I don't know, and you have to just be okay with, you know what, I don't know everything. I'm going to call on a friend to help me.
That's a really excellent point, and I'm glad you brought that up, because I think a lot of times folks, myself when I was younger, I thought that scientists were super geniuses who knew everything, when in fact, generally they know a lot about one thing. Because you can't know everything. It's just not possible. So being a scientist is achievable, it's doable, it's not scary. And yeah, also in 4th and 5th graders, you are not too young to learn to code. You can totally start learning that now. It'll come in handy later. We've got a question from Ms. Camacho's class. This is a fun question. I love this sort of hypothetical sort of question. Is there any way that we can breed pugs, like the dogs, using what we know about their DNA so they can breathe easier? What a good question.
That is a fantastic question. I honestly, going off of Elizabeth's statement just before, I don't know. Maybe we gotta find out from someone who knows dogs better. Yeah, I'm not sure.
Well, that's crazy because I actually foster pugs. And so I know a lot about like, oh, of course you have to have pugs get the surgery. They get a little nose job so they can breathe better. I think if we had like all the resources in the world, we'd want to take all the pugs ever that can breathe normally and be able to just breed them together. And through the natural selection, through being able to see which pugs survive because they can breathe better, we could create a special breed of pugs that can breathe.
I love that hopeful answer. I bet we could. If we put enough resources to it, we could do it. We've got a question from Melanie here. Does Illumina have any outreach program for high school students during summer, like an internship program or anything they can get involved in?
Yeah, we do. And I think it's open right now if you want to apply. But I do believe it is only for college students, not for high school students. But eventually, when things get a little bit better for people who are local, we do tours where you can come to our campus and check out our labs and stuff.
Mm-hmm. Yeah, and the program's called I Aspire, but I know there's definitely programs out there for high school students where they will take you to different medical fields, whether that's touring hospitals, touring biotech companies. So definitely if you look for the internships or the programs, they're out there.
Awesome, thank you so much. We've got a question from Megan. Does your company invent the tools that you use?
Yep.
Yeah.
So in our in our lunchroom, our cafeteria, we have this huge wall of patents, which is basically like the paper saying, hey, we made this and we invented this. And so, yeah, a big part of Illumina is making and inventing these sequencing machines, as well as all of the solutions that go into them. And that's how Illumina is sort of this big powerhouse in the biology industry, because we make these incredible machines that so many other companies depend on.
That's for sure true. Yeah. Thank you. The next question is from Alexa. How frequently do mistakes happen in science, and are mistakes encouraged or generally frowned upon?
They happen all the time, and some of the inventions came because of mistakes. So yeah, I mean, I don't know if I would say they're encouraged, but don't be discouraged if it does happen, because that's always something that we can learn from. It's something that we like, okay, I shouldn't do that next time because this happens, or it's like, oh, well, this happened, so maybe we can take it another step further and see what happens after that. So yeah, I mean, mistakes happen all the time, but it's not a bad thing.
Mistakes have produced some of like the biggest accidental world-changing things in science, like penicillin, which was the first major antibiotic found by mistake, and so many others, like GFP. Folks at home, I don't expect you to know what GFP is, but it's basically a way that we can make cells light up for us. And in science, it is huge. We use it all the time. And it basically, if you've ever seen a picture of cells in like bright colors, like green and red and orange and blue, we would not have been able to take that picture without somebody studying a jellyfish and stumbling upon this protein. So mistakes in science sometimes teach us just as much as when we do it right. So yeah, across the board, they're useful, even if we that day in the lab wish we hadn't done it. Overall, sometimes they're really, really helpful. The next question is from Tiana. There seems like there are so many jobs in biotech, understatement, big time. Which jobs do you feel like are super cool and growing, but maybe not super well known by people?
Ooh. Well, so for me, what I think is really cool is how in biotech, we're starting to introduce like automation. And so there's automation engineers and automation biologists that are getting to know more about automation. And they use these giant robots that do all of like moving the liquids that we do manually. And so you have to learn how to code for that. And you basically are coding this giant robot to do science for you, and then you can put that on a sequencer and find out the answer there. So I think that's pretty cool. Going back to Sarah saying that it's never too young to learn how to code, so that you guys can work on that as well.
Awesome.
Yeah, I have a friend and she works in biophysics, but basically she dives underwater and she goes to extreme environments and she researches the bacteria that can live in those extreme environments. And basically she can compare the fact that if this bacteria can live in this really extreme environment in the ocean, what can live on Mars? What can live on planets? And so she's able to connect like space science to stuff that we have on our planet right here, which I think is just crazy.
Awesome. That's super cool. Amazing. We've got another question. This is from Brielle. What has so far been your favorite moment in your career?
Okay, I'll go. So I work on something called NIPT. So it's non-invasive prenatal testing, which is just a fancy way of saying checking on if you're pregnant, if your baby is okay, or if there are going to be some anomaly, there's some changes in their DNA. And so last year I was pregnant and I had to go get the NIPT test, and it was the test that I was working on. So I got to use what I work on at work, which was super rad. So cool seeing like Illumina on the bottom of the results.
That's awesome.
I think it might have been the full circle moment when I went into the Illumina lab the first time. And I had never seen those machines before in real life. Like I had only seen them in textbooks, or, you know, we would whisper about them in school. So I think walking into the lab for the first time and like being able to touch the machines and put the chip in was very surreal for me.
Awesome. Very cool. We've got a cool question here from Ms. Camacho's class. Have you ever seen a radioactive animal?
I have not.
There are some tests, although I admit they're kind of phasing them out a bit, but they use very small amounts of radioactivity to do tests of various kinds. Like we call them assays, but assay is just a fancy word for like an experiment, a test of some kind. And back in the early 2000s, and even when let's see, this was in 2013, we were still using radioactivity in the lab. And for some experiments, some parts of science, it's essential. But we've found ways of not using radioactivity because there's a whole room that's like the hot room, like the room where the radioactivity is, and you have to wear a special lab coat and you have to wear a little like band on your arm to make sure that you're not being exposed to too much radiation. It's just a little spooky. It's a little scary. And so we've generally phased it out. But everyone is a little tiny bit radioactive. A banana is slightly radioactive. So in a sense, we're all radioactive animals, but not like Chernobyl radioactive. For those of you who don't know, Chernobyl was a nuclear disaster that happened a while ago. Anyway, I digress. Let's see. We still have a lot of questions here. Could you give us, so you mentioned that you had previously worked on COVID tests. Could you tell us just a little bit more about what that process was like and how you even do that?
Yeah, for sure. So I was a lab tech, which meant that I saw the samples firsthand, and we use a test called PCR, the polymerase chain reaction, to be able to determine if that sample is positive or not. And so we usually had 3 different phases to be able to complete that. First, we had the extraction phase, which means I would have to, take out the goop from the tube and put it into a separate plate, and we would extract the DNA and the RNA from the sample itself, because you can't just test a sample directly. And then we would put them in Kingfisher machines. After that extraction process, we would put them in a huge amp machine, which basically, when you have DNA, you only have a little bit of it. And to be able to read DNA, you need to make a ton of it. So this machine will make this DNA super massive. And then when it's that big, we can finally analyze it and see and read the DNA. Is it positive or is it negative for coronavirus? And so that's what I did.
Awesome. We've got a question here from another class. What kind of genetically modified animals do you work on? Do you work on any at all?
I don't work on any genetically modified animals. We mainly work with, I think, human DNA.
Certainly, though, Illumina technologies are used on samples from genetically modified organisms like 1,000 times a day, like 1,000 times a day. So genetically modified animal, that sounds sometimes to some people like pretty wild, but almost every mouse that you have in a lab, about half of them are going to be genetically modified. And so sometimes a scientist will have like a tweak in the DNA that changes how they do something. If it's cancer related, maybe something's tweaked to give them more cancer so that they can study what happens in cancer. Maybe immunologically, so in their immune system, they do something different or whatever. And a lot of times scientists, let's say at Harvard, at Boston University, at Drexel, they'll take a sample, but they won't have the sequencing stuff in their lab themselves, and they'll send it to Illumina. And so do they physically have the modified animal? No, but they have the DNA from the modified animal they're working through all the time. And so genetically modified animals are all over the place in science, but they're not they're not as spooky. They just look like a mouse that's a little bit different inside. So yes and no, I guess. We got a question here from Amanda. When you tweak part of DNA, what does that mean? Are you changing the chemistry? Are you inserting something into it? Is it similar to computer coding? Do you two want to answer that, or
it's all of the above, depending on, and like we said with the GMO animals, it could be we take the DNA and we insert it into a plasmid and just test that. It could be that you change something about the chemistry of the DNA to see, to be able to perform the test on it. What else is there, Sarisha?
Sometimes you can cut a little chunk out, like if you're like, what would happen if this animal didn't express this? And then you use proteins that are specific to playing with DNA. There are like certain protocols that you can use for different animals that do different things. And one of those things is just cutting it out, and that's called a knockout. It just depends, right?
Depends on what answer you're looking for.
That's exactly right. Thank you. I think we have time for one more question before. Oh, this is a great question. This is like very foundational to Illumina. How do you read DNA?
So when we have our sequencing machines, we do use GFP or different fluorescents to be able to read it. And it's because DNA is made of 4 main parts: an A, a T, a C, and a G. And so we label each of those letters in the DNA with a different fluorescence color. And through, we have like a flow cell or a little plate, and when we run it through the sequencer, we read those different fluorescence colors, and based on the pictures that we take, we'll be able to build the sequence. Oh, if it's green, red, blue, red, red, then we know what those letters mean.
Yeah, basically color code the bases. There are only 4/5, and then the colors tell you what it is. And you get you get this file that's just like letters and letters and letters and letters after each other. And that's how you read it. Awesome. So thank you both for joining us today. This was super, super cool. We learned so much about working in biotech, and we like to ask everyone the same 2 questions at the end of these sessions. Here's the first one that we'll give to both of you. If you had everyone's attention in the whole world, and you can tell them one thing about your area of expertise, what would it be?
probably that you can do it too. You know, like it's not just me, not just Risha, like really anybody can do science, anybody can put in the work to really change the world.
Yeah, I think it agreed with what Elizabeth said. It's just as long as you're willing to learn, you can do it too, and it's super cool. And there's still so much more to learn. So don't think that we figured it all out and there's no need for any more scientists. There is so much we don't know, and everyone can help us learn more.
That's a great point. I remember being in middle school and reading the textbooks, and I was like, well, I think they got it. Like, what else is there to know? And the deeper you go, the less we know. It's wild. The last question for you two before we wrap up today, you still have everyone's attention in the whole world, and you can tell them one thing about anything. It can be related to biotech or related to pugs. It can be anything. What do you tell everyone?
Just have fun. Don't take anything too seriously.
Definitely try things that scare you, whether that's coding, whether that's skateboarding, rollerblading. You need to try things that scare you.
Solid advice. All right. Thank you both. Kay, thank you for interpreting for us. Thank you for you both for all of your insight and your knowledge. This was super cool. For everybody at home, thank you for coming. This was super fun. We are meeting again next week. We're going to be talking to a scientist who works on not just jellyfish, healing jellyfish. So she studies how jellyfish heal themselves and what we can learn from jellyfish. We already learned today that we learned about GFP, one of the most foundational like biotechnology things ever from jellyfish. What else can we learn about jellyfish? A lot. So come join us. That'll be on October 20th at 1 p.m. Eastern. We hope to see you there. And thanks for coming. Have a wonderful rest of your week. Thank you both. Thanks.
Bye, everyone.
Bye.